Let's review the operation of the atrioventricular valves, and then we'll talk about the operation of the semilunar valves, right? The atrioventricular valves are the tricuspid valve and the mitral valve, also called the bicuspid valve. So the first thing that's going to happen is when the ventricles relax, the atria are contracting, right? That means when the atria are in systole, the ventricles are in diastole. And when the atria are in systole, the pressure in there is greater than the pressure in the ventricles. So the blood literally pushes open the tricuspid valve and the mitral valve. So even though this picture kind of looks like the papillary muscles are pulling open the tricuspid valve here and the mitral valve over here, they're not doing that. The blood is pushing its way through, just like we would if we were pushing open swinging double doors. Now, what closes the atrioventricular valves? The blood does. So after the ventricles have filled, then they go into systole while the atria are in diastole. So now the pressure on blood inside the ventricles is greater than the pressure on blood in the atria. So the blood wants to go backwards. So it pushes back against the door it just came through. And when it pushes backwards, it actually shuts that door. So you can see here, the blood has shut that door. And shutting that door is going to allow pressure to build up inside of the ventricles. Pressure builds up, builds up, builds up until it's big enough that ventricular systole will cause the semilunar valves, the pulmonary valve and the aortic valve to open. So when the ventricles contract, they're going to go ahead and push open the pulmonary valve and the aortic valve. The closing of the tricuspid and the bicuspid is a little bit before the opening of the pulmonary valve and the aortic valve. And then blood goes out into there. Now, when the ventricles go into their own diastole, when they are relaxed, then the pressure inside of the great arteries is greater than the pressure inside the ventricle, because there it drops down to zero. So that means the blood will come rushing backwards, and it ends up pushing back on that set of one-way swinging doors, and that slams them shut. Because the valves are designed so that they are not allowed to swing both ways, right? As one-way no, that doesn't look right. As one-way doors, they go this way, and then they go shut. This way, and then shut. They're not designed to go this way and then that way. They don't do both ways. They're one-way doors. Which brings me to another important concept, that the reason that the atrioventricular valves each have got chordae tendineae attached to papillary muscles is not to open them up. Opening them up, someone else does that. These guys are designed to protect that set of one-way doors so that they do not prolapse. They do not prolapse. So what would prolapsing be? So let's look right now. We're looking at the bicuspid valve. So it's got two doors, and it is designed in this case to open up this way and to get shut. Open up this way and get shut. So blood comes into the ventricle from the atrium, and then when the ventricle contracts, it shuts the door. Valvular prolapse would happen if the chordae tendineae rupture, if they break. Then what would happen is the valve would open up inward, but without the chordae, then when the ventricles contract, they would open up the opposite way. And now, every time the heart beats, half of the blood would go forward, half of it would go backwards, and that's not compatible with life. Okay. So now we're going to start talking about the electrical activity of the heart. Up until this point, we've been thinking about the heart as a right side and a left side. The right side pumps blood to the lung, the pulmonary circulation. The left side pumps blood to all the rest of the body, the systemic circulation. But now that we're talking about the electrical activity of the heart and its contraction, I'm going to ask you to think about it as a top part and a bottom part. The top part is all of the muscle of the atria, and all of the heart muscle cells in both of the atria they're all connected to each other, and so they form an organization that's called the atrial syncytium. Also, all of the cells of the ventricles, well, all of the heart muscle cells, the myocardial cells of the ventricles, they are all attached to each other in a clever way, and they make up what's called the ventricular syncytium. So if I say the word atrial syncytium, I'm not talking about something that happens. I'm discussing the organization of the heart muscle cells. Now, we're going to talk about how the heart contracts. And it's important to know that the heart is very different from skeletal muscle. Skeletal muscle is designed to contract when a somatic neuron releases neurotransmitter, and that neurotransmitter will say, hey, contract, and then the skeletal muscle obeys. It's not the way heart muscle goes. Heart muscle is different. Heart muscle cells, they will set up their resting membrane potential, and then they instantly allow positively charged ions to slowly leak into the cell. By allowing positively charged ions to slowly leak into the cell, they're just slowly bringing themselves up to that moment of threshold, and then they'll contract. So they don't need anyone to tell them to contract the heart. If you took the heart out of a person, as long as you kept the heart cells alive, it would go ahead and keep on beating. You could get it to beat as long as you wanted to, as long as you kept all those heart muscle cells alive. So these autorhythmic fibers, they set up a resting membrane potential, but then they allow positively charged ions to slowly leak in, which slowly brings them to threshold in the moment that they contract. And that is why they are described as being self-excitable. They repeatedly generate the action potentials that will end up triggering the heart contractions. Now, the fibers of the heart that allow positively charged ions to leak in most quickly are found at two places, the sinoatrial node and the atrioventricular node. The sinoatrial node, those guys leak super fast. So they're constantly getting to their moment of threshold really, really quickly. And when they do, they end up causing a heartbeat. And so those cells are described as the pacemaker of the heart because they set the pace of heart contraction. So let's talk a little bit more about the structure of heart muscle cells, myocardial cells. They're very short. They're not long and tubular the way skeletal muscle cells. They're short and they've got all kinds of little branches. And each of those little branches allows them to connect to 6 or 7 of their friends immediately around them. How are they connected to their friends around them? Through these things called intercalated discs. Intercalated discs. Now, the intercalated discs do a couple of things. Yes, it's true that they actually attach them together. So it's like they're holding hands, right? If one of them is going to contract, he's going to drag his friend with him. So that's one of the things the intercalated disc does. But we're not going to be talking about that very much. We're going to be talking about these things called gap junctions that connect the cytoplasm of one myocardial cell with the adjacent myocardial cell. Let's look at this image down here on the left. If this cell was leaking and allowed positively charged ions to leak in, it would get to a moment of threshold, and then it would open up all kinds of channels and allow a lot of positively charged ions to leak in. And those positively charged ions would leak over to that cell, and leak over to that cell, and leak over to that cell, and leak over to that cell through the gap junctions in the intercalated discs. And so when this guy contracts, then his friends here and here and here and here, they'll also get to threshold and they will contract. So what is the atrial syncytium? Please remember, it's not something that happens. It's not an event. It is our way of describing the way all of the myocardial cells of the atrium are connected to each other in a clever way, and all of the myocardial cells in the ventricle are connected to each other by these intercalated discs. So if you look at these two pictures, are these two pictures the same? Well, they're both crowds of people. But the one above, you would recognize as a band, right? And this one down here, well, that's just a crowd of people. Well, how are they different? Well, this one, when one does something, it causes all the rest to do something. This is like a syncytium, okay? This is like heart muscle tissue. if this guy were to do something like turn right, then that guy's supposed to do it next, and then him, and then him, and then him. And you get this organized effect. That is the way heart muscle is organized. Skeletal muscle is not organized that way. If this cell contracts, this cell does not care. That's more like just a crowd of people. If this guy decides to take off and go get some popcorn from the vendor, it's not going to make the people around him do anything. Right? So the cardiac conduction system, it's really important that remember that the cells that are going to initiate the action potential and the contraction are the heart muscle cells themselves, which is why it's described as myogenic. Myo means muscle, genic means originating from. So the heartbeat originates from the muscle cells of the heart, not a nerve cell telling it what to do. And it's autorhythmic. Even if you take one heart muscle cell out of the heart and just put it in a petri dish, periodically it will contract because it has its own rate of leaking and it has its own rate at which it's going to arrive at that moment of threshold, have an action potential, and after the action potential, it is going to start a contraction. Right? We will pick this up at the next video.